Happy Tuesday, back with another video. So now I'm doing a ligation on the linearized plasmid from last week and the fragments from the genome. So this means that I'm putting the fragments, the genes, inside the plasmid and ligating them to one circular piece of DNA. Preparing some media to plate some colonies out. Still have to sterilize them in the autoclave. The autoclave is filled and ready to be put on pressure. Now the pressure will build up to 15 psi, meaning it will be very hot inside and all the bacteria will be killed. The autoclave and the ligation is still running. Let's see it. Autoclave has been running for 20 minutes. Let's turn the heat source off and let it cool down, and the pressure will go back to 0. Autoclave is cooled, so I can open it. And all of this is now sterile. Alright, here I have my competent E. coli cells in which I'm going to put the ligated plasmids in. So that's the next step. Do heat transformation to put the plasmids inside the bacteria. Then these bacteria will multiply, so they divide. And when they divide, their DNA will also be copied, including the plasmids. So that's a way to produce the plasmids. Let's do that. A heat shock. Right, the plasmids are ligated, so the genes should be in the plasmid now. Let's transform the E. coli with those plasmids. So this is now my plasmid solution.
Here I'm adding the plasmids to the E. coli cells. Next, the heat shock is performed by exposing the cells to 42-degree water for 45 seconds. In this step, the plasmids will enter the E. coli cells. Then the cells are incubated for 1 hour in special media so that they can recover from the heat shock.
1 hour later.
In the next step, I will plate the bacteria containing the plasmids on some plates I just made. Quick after work pool workout. I just cleaned their filter here. I changed the position also just to see the difference. Look at the female. Imagine this frog being fluorescent, red or green. I'm planning to do the experiment the week I'm posting this video actually. Yeah, I'm very excited to do it. Happy Wednesday, and for once I don't have to go to the other campus 2 hours away from here. I can just follow it online because for the first lecture we had a meeting because we have like a big task for biomolecular nutrition, and that's already done, so now I'm doing something else. And then the other 2 classes in the afternoon I can follow online. That's awesome because I can save a lot of time not traveling. Thursday, 9 AM, back with another day in the lab. Let's see if they have grown. Alright, my colonies have grown. Very nice. So this means that the bacteria should contain the plasmid because they grow on the media with the antibiotic. Only bacteria with the plasmid should be resistant to. But now, how can I check my colonies if they contain the plasmid? Just as last week with a colony PCR. So that's probably going to take the whole day. And then at the end of the day, I know if my colonies are positive for my new plasmid with the additional gene inside.
Here I'm going to pick 8 random colonies of each plate and mix the cells in a drop of water. Each plate should contain bacteria with a new plasmid. Then the cells are microwaved to break them open and expose the DNA. Next, I'm distributing each DNA water solution over 2 tubes, because I want to do 2 PCR reactions for each sample, one control and one for the plasmid. In the clean room, I'm preparing the master mixes, which contain all the ingredients to make the PCR reaction happen.
Master mixes are ready, then the final step is to add 20 microliters of this to the 5 microliters of DNA of the colonies, and then I can put the PCR reaction on in the heat block. Alright, 1 hour 15 minutes left. Let's already make my gel. Because I'll have to put it on gel after the PCR reaction, of course. This is the gel red, which will mix with the gel solution. And this binds the DNA and causes it to fluoresce red under UV light. Then my agarose is dissolved in the buffer, and let's add it to the mold. Alright, and the gel is ready. It only has to solidify. Then I can use it. But first, lunch. PCR reaction is done. The gel has solidified. And I already mixed some PCR reaction with loading dye. So those are all ready to be put on gel. And we can run it and visualize the DNA fragments. And then I finally know if the plasmid were in my colonies to begin with or not. Okay, bad news here. The upper layer you can see the fragments are as long for the positive control as for my colonies, so that's not good news because it means the plasmids have transformed inside the E. coli, but it are plasmids without my fragment inside. My gene of interest of the bacteria is not inside the plasmid. So tomorrow I'm gonna pick 8 different colonies and just check to be sure if they contain a plasmid with my fragments inside, and otherwise the ligation or something has failed. Thus tomorrow I'm gonna have to do exactly the same as today, but just on 8 different colonies, because the ones I checked did not contain the right plasmid. It was a hell of a training session. Alright guys, 9 AM, let's do exactly the same as yesterday, but on not 8 but 12 different colonies. I already marked them here, for example. I'm gonna check 12 different colonies and hopefully they contain the plasmid with the fragment inside. Let's do it with the colony PCR. I'll skip to the part of the gel because otherwise it's just the same as yesterday.
Later.
Moment of truth, it's 3 PM now. I've worked the whole day on this colony PCR of 12 additional colonies. Let's check if any of them contain my fragment. Yes! Even 2 of each one. This one And this one, colony 9 and 11, and here also 2, this one and this one. Awesome. Only 2 of them contain the plasmid with my gene inside, ligated successfully. Very happy. Quite lucky that I picked those colonies. So for all the 20 colonies of this one, only 17 and 19 contain the exact right plasmid, and of the other one, colony 16 and 19 contain the plasmid with my gene ligated inside. All the other ones just contain the regular plasmid without the gene of interest. The last thing for today is to pick the colonies that were positive, the 4 ones, and put them isolated on a new plate and grow them for the weekend. Happy Sunday! Back at work!